Here you can see our mind map. In our mind map, we focus on two different kinds of lung fibrosis, CF and IPF. Idiopathic pulmonary fibrosis is a type of chronic scarring lung disease characterized by a progressive and irreversible decline in lung functions. Symptoms typically include shortness of breath, nail clubbing, and dry cough. Complications may include pulmonary hypertension, heart failure, pneumonia. The cause of FPI is unknown, but certain environmental factors and exposure have been shown to increase the risk of getting IPF. Cigarette smoking is the most accepted risk factor for IPF. Also, exposure to several types of dust and viral infection may be associated with IPF and other fibrotic lung diseases. However, nowadays there's still no cure for IPF. The treatment now is to slow down the process of fibrosis and thereby extend the life of the patient. In our challenge, we will focus on the cause of IPF. The injured lung tissue activates the TGF-beta pathway, which activates cytoplastic SMAD proteins. Thereby, SMAD2 and SMAD3 get phosphorylated by the TGF-beta receptor type 1, and they will form a complex with SMAD4. This complex regulates the nuclear expression of different types of genes that have effect on the fibrosis. The research question for our challenge is as followed. Can the inhibition of the activation of SMAD3 disrupt the development of fibrosis in IPF? CYS3 is a very potent and specific inhibitor of the phosphorylation of SMAD3. We expect that CYS3 will prevent the phosphorylation of SMAD3 and thereby the binding of SMAD3 with SMAD4. This in the end will prevent the fibrosis in IPF. To test our hypothesis, we formulated the experimental setup depicted here. First, lung tissue was cultured from both healthy and diseased donors. All 5 cell cultures will undergo the same experiment. qPCR will be used to determine the amount of gene expression of 2 gene collagens type 1 and 3. Next, the quality of our SMAD protein is determined, allowing us to compare the concentration of these proteins. Finally, the different collagen tissue is stained and observed. By comparing the results of the qPCR and immunocytostaining, a conclusion can be drawn concerning the effectiveness of inhibiting SMAD3 to disturb fibrosis in IPF.